Hello friends, in this video I'll talk about wheat, its cultivation, breeding and activity of the pH locus. Wheat is a prime most cereal crop of the world consumed by more population in the world. The origin of wheat, first cultivated wheat occurred about 10,000 years ago as part of the Neolithic revolution. Cultivation spread to the Near East 9,000 years ago. Hexaploid bread wheat made its first appearance. The main route into Europe was from Greece, Balkans, Italy, then UK, and then it spread to across the continents and across the world. It is a food for 30% of the world's population, rich source of nutrients, rich in protein, carbohydrate, many vitamins, minerals, etc. There is a need of increasing productivity and increasing the population. Climate change are the both two challenges ahead to wheat. Exploring the genome content to understand molecular basis for agronomic trait is also a major aspect in wheat improvement. The reasons for low production and productivity of wheat at the time of Green Revolution was tall growing plant habit resulting in lodging when grown under fertile soils, poor tillering and low risk capacity of the varieties used, high susceptibility to disease and higher sensitivity to thermo and photo variations, etc., resulting in poor adaptability. Classification of wheat are Triticum, Aestivum, Durum and Dicoccum. The share of production of Aestivum is 95%, grown mainly in India in the parts of UP, Punjab, Rajasthan, Bihar, Madhya Pradesh, Jammu and Kashmir and Assam. Triticum durum contributes 4% and this is grown in Madhya Pradesh, Maharashtra and Gujarat. Third one is Triticum dicoccum, cultivated and contribute 1%, grown in Karnataka, Maharashtra and Tamil Nadu. The key events in evolution of wheat: two hybridization and genome doubling events, that is diploid into diploid gave allotetraploid, AA into BB gave AABB, then diploid was crossed, that gave allohexaploid ABD genome. Chromosome doubling may have occurred before hybridization. The genome contribution species are: first one is cultivated iron corn wheat as AA. The AA genome was contributed from Triticum urrutu. Then BB given by Agelopsis crc, Agelopsis peltoides. Then the combination gave AB by chromosomal doubling. Wild tetraploid wheat was Triticum turgidum. That was cultivated 10,000 years before Christ, and emmer wheat is AABB. The DD was taken from Triticum toshi, that gave ABD, and from there by doubling, chromosome doubling, hexaploid wheat, that is Triticum aestivum, evolved 8,000 years ago. The evolution of wheat, examples of spikes and grains. This is the kind of spikes and the variation found in the spikes appearance. Urrutu is somewhat in conical shape, then turgidum somewhat ovoid or spreading type, then aestivum is round shape, that is just like the cat's tail, then toshi is just like the teosinte maize, the primitive cultivar of maize, then spelta is somewhat blackish in color. Then monococcum and cerci are given at the adjacent sides. This is the overall evolution, how the Triticum aestivum and the other progenitors of wheat came from ancestral diploid species Triticum toshi and the AA genome progenitor and Cytopsis section, that is SS progenitors originated, that was divided into Agelops bicornis, crc, sharonensis, longicima and speltoides. From A genome, Urartu and Monococcum evolved, and from there A genome crossed to Erythranium, that became AAGG genome combination. Then when it combined with Turgidum, it gave AABB. The AAGG was Triticum timofevi, and finally it gave birth to Triticum zhukovsky. The other combination is Turgidum into Toshi, that gave birth to Aestivum. The domestication led to cultivated wheat before 10,000 years. Coming to bred wheat, that is Triticum aestivum species aestivum. The chromosome number is 42. Wheat has three homologous chromosome sets A, B, and D. Disomic inheritance preserves hybrid nature, behaves as a diploid at meiosis. How it behaves, that will be addressed in the last or end slides. The meiotic behavior, how even though the genomes are different, but the homologous pairing will be directed towards homologous pairing, and normal segregation pattern will be obtained even it is a hexaploid. This is the pairing pattern. Coming to the Green Revolution aspect. Wheat was a major contribution for Indian as well as worldwide Green Revolution. Important scientists like Salman, Norman Borlaug, Swaminathan contributed greatly for Green Revolution. Norman Borlaug was a pathologist, and he is a major contributor for Green Revolution who worked with Mexican wheat varieties. Many of the cultivars brought from Japanese type or Japan during the World War contributed majorly for developing the semi-dwarf wheat variety. Scientists or the persons brought Japanese semi-dwarf Daruma and gave it to Borlaug. Then they crossed it with US winter wheat having high yielding ability, Fulls, that gave Fulls Daruma cross combination. This was semi-dwarf and high yielding. From there they crossed it with Turkey Red. Turkey Red was US winter high yielding variety that gave birth to Norrington. Norrington is a semi-dwarf, winter and high-yielding variety. Norrington was again crossed to local adapted US network that gave Guinness. Guinness series was semi-dwarf, winter and US adapted. Again it was crossed to local strain, and the new wheat varieties were semi-dwarf, high-yielding, adaptable, rust-resistant and fast-maturing spring type. The major objective of the entire crossing program was in order to bring the semi-dwarf mature with high yield, adaptability, and resistant to different diseases. The Green Revolution attempt started during 1943 in Mexico. Mexico took around 15 to 17 years to attain the Green Revolution, but in India it only took 5 years. The government of India appointed commission in 1961 to assess the feasibility of increasing crop productivity under prevailing Indian ecological conditions. Several policy decisions and actions were taken by government time to increase the production and productivity in the country. The Ministry of Agriculture, Government of India, has launched many programs like IADP, then frontline demonstrations, mini kit demonstration program, state level training program, and different workshops and seminars in order to popularize the introduced wheat varieties, semi-dwarf wheat varieties, Sonora 64, Lerma Rosso, Choti Lerma, and all those things. Even the secondary introductions like Sonalika and Kalyan Sona. The major problem when the semi-dwarf cultivars introduced from Mexico to India, the seeds were having dark red or reddish tinge color, and that was not preferred by the farmers because of its stickiness and the poor chapati making quality. After that, from there, they did selection as well as the secondary introduction like Kalyan Sona and Sonalika. After that, by introducing the combination of 2 to 3 dwarfing gene, made revolution in the wheat era in India. Additional technologies recommended for good cultivation of wheat were zero tillage technology, general instructions and precautions while doing zero tilling, the machineries supplied to the farmers and subsidies given for that the particular machineries buying, then furrow irrigation, raised bed planting system, rotary tillage technology, all those things. Wheat, coming to utility of wheat, wheat contributed to different food products like chapati, pasta making, bread, biscuit, noodles, cookies, dalia. Industrial uses like gluten is useful in preparing coatings, polymers, etc. Wheat starch is used in cosmetics, paper, pharmaceutical industry. Other uses like straw and bran as cattle feed. Food value, carbohydrates is 70%, protein 12%, fat 1.7%, minerals 2.7%, fiber 2%, and moisture 12%. The botanical description of wheat. Wheat has bifid feathery stigma. This is a peculiar character with 3 stamens or anthers, and it is a hermaphrodite flower. The botanical description or biology of wheat. The flowers are bracteolate, sessile and bisexual. Stem are cylindrical, distinct node and internode, herbaceous. Leaves are alternate, sessile. Leaf base tubular sheath open surrounding the internodes completely. Linear, parallel venation. Inflorescence is sessile or stalked. Each unit is called spikelet, panicle of spikelets. Root is adventitious, fibrous and branched. Culms are usually they grow 1 to 2 feet tall and less than even 1 feet. They are terminated with inflorescence called panicle. Thus, wheat plant forms crown at the soil surface from which a fibrous root system penetrates the soil. The major diseases of wheat are bloom blotch, leaf blotch, rot, pythium rot, rust. Rust is the most devastating disease in wheat. Then sclerotial disease, diplophorus leaf spot, leaf blight, foot rot. This is the leaf rust. The major disease in India is controlled by the deployment of leaf rust resistant genes. Many of the attempts are made in order to overcome the rust problem in India, like gene pyramiding, marker assisted selection, gene deployment. One major aspect is gene deployment, which prevented the Puccinia path traveling from south to north. The different region gene deployment is nothing but geographical location of the different genes for specific disease. Here in Himachal Pradesh, they deployed HS365. These are the varieties and the deployment of LR26 plus other genes. Then in the same, they in the other variety they deployed LR23. Then in HPW42 they deployed LR26, LR1, LR34 and all. Likewise, in different regions they incorporated different genes for leaf rust in different zones. Coming to pests of wheat, one is termite, that is Odontotermis obesus or Macrotermis obesus. Then stem borer caused by Sesamia inferens, Guggia weevil, thrips, armyworm, wheat aphid, surface grasshopper and cutworm. Harvesting and threshing in wheat. The crop harvested when the grains become hard and the straw becomes yellow, dry and brittle. Most of the harvesting in India is done with sickle. However, in recent years, some states like Punjab, Haryana, Uttar Pradesh, harvesting and threshing is done by combined harvester at large scale. Maturity time of crop differs from zone to zone, and so it is the harvesting. Coming to wheat genomics, the modern bred wheat hexaploid has 42 chromosomes. The genome size is 17 gigabase. Then more than 80% repeats, 2% coding sequence, high sequence similarity within subgenome ABD. The wheat genome was sequenced by IWGSC, that is International Wheat Genome Sequencing Consortium. The global landscape of endosperm gene expression. The endosperm composed of three main tissues. Various tissues were analyzed from different developmental stages. Around 85,173 total genes were found in hexaploid wheat. The equal contribution of number of genes from all the three genomes were found. Then preferential expression of gene based on tissue and similar expression between subgenomes. These all results were obtained from wheat genome sequencing. The complexity of genome. Highest complexity is found in hexaploid wheat, followed by tetraploid, followed by diploid. The complexity or genome size of hexaploid wheat is 17 GB, then diploid wheat is 6.2 GB, tetraploid is 6.2 GB, and the diploid, that is different species, Triticum monococcum and ururtu, are 5.7 GB, whereas Aegilops peltoides, Sharonensis and Toshi are having 4.9 GB. These are diploid species. Coming to the breeding methodologies, pedigree, intervarietal hybridization, winter into spring wheat crosses, mutation breeding, aneuploid and genomic breeding, restructuring plant architecture like semi-dwarf wheat, shuttle breeding and photoinsensitivity, hybrid wheat, apomixis, functional genomics and molecular genome breeding are the major wheat breeding aspects. Coming to hybrid wheat, in wheat improvement program only specific methods are incorporated or utilized because of the genome complexity, and the success rate is quite less, even though many of the techniques and methodologies are utilized in it for the improvement. The hybrid wheat is quite unsuccessful because of the planting habit, the flowering habit, as well as the hexaploid wheat itself is a hybrid, so exploitation of further hybridity is almost less, that is only 30%. So the hybridization program in wheat is very low or in infant stage. Coming to the homologous pairing, as in the initial stages I talked how the ABD genome in wheat give normal pairing and further propagation. This is because of the pH gene suppression. Initially, it was assumed that three diploid species whose genome had gone to make up hexaploid wheat were strongly differentiated. In 1952, it became clear that the corresponding chromosomes of three different genomes are genetically very closely related. Riley and Chapman in 1958 discovered that homologous pairing is suppressed by a gene or genes on a long arm of chromosome 5b that became known as PH1. PH1, led by Professor Graham Murray, research was on wheat meiosis and the PH1 locus. The effect of PH1 gene leads to either multivalent formation or univalent. It is critical in maintaining the genome stability in wheat. Effect of PH1 locus suppresses pairing between related chromosomes. That means it leads to homologous pairing if wheat and rye hybrid. Due to the suppression of PH1 gene on the long arm of chromosome, the wheat 3 genome complexity is unlike understanding type, but if the wheat is crossed to some other related species or related genera, like in wheat rye hybridization system, because of this pH gene the crosses will be unsuccessful. For this, understanding this homologous or homologous pairing is very essential. So whenever the pH1 locus is deleted, pairing is induced between homologous chromosome. So, what is pH1? The wheat genome is very large, that is 17 GB, 3 closely related genomes. No natural variation in pH1 phenotype. It can't create segregating populations, the starting point of all previous positional cloning projects. But X-ray and fast neutron irradiation do that. A single deletion of pH1 of the locus gives 70 MB size. That means pH1 occupies 70 MB size in the genome. Deleting pH1 increase the CDK activity, which increase histone 1 phosphorylation. The result of increasing the CDK activity and increasing histone H1 phosphorylation is pairing between homologous chromosome that will enable the formation of triticale or development of triticale by wheat into rye hybridization. pH1 forms bivalent by eliminating multivalents. This is at the cytogenetic level or at the pairing level, where the normal pairing and crossing over or recombination were observed during synaptonymal complex formation or zygotine or Pachytene stage. Like this. At both these stages, condensation changes occur which would be affected by histone 1 phosphorylation. Increased histone 1 phosphorylation will be observed when pH1 locus was deleted. What happens at homologous recognition stage in wheat? The identical chromosomes zip up from their telomeric region when the pH locus was deleted. That leads to homologous recognition or homologous pairing in the wheat. This is a study in wheat chromosome remodel in both the presence and absence of pH1, but there is synonymous chromatin remodeling in the absence of pH1, correlating with more incorrect associations at homologous recognition stage. This is synopsis, how synapsis and recombination happens in the dividing cells. When adjustment without pH1 means the proper recombination will be done, but when chromosome segments forming a circular structure means with the little synaptic adjustment with pH1 means that leads to no recombination. This is how the chromosomes in the synaptonymal complex lead to adjustment or pairing with each other. This is in presence of pH1, where chromosome cannot pair due to their length, and in the absence of pH1 synapses happen. pH1 effect is important agronomically. How? Because wild species of wheat carry important traits for disease resistance, salt, cold and drought resistance. Wheat is a global crop with a complex evolutionary history which gave it its hexaploid status. pH1 stabilizes the wheat genome by controlling pairing and effectively turns it into a diploid. pH1 is related to human CDK2, which phosphorylates histone 1 and modifies chromatin conformation. pH1 could be used to introduce novel genes into commercial crops. Switch pH1 on and off in a light wheat varieties crossed with wild species to introduce novel gene to the commercial crop. That's why I spent so much time in order to explain the process or the activity of pH gene in wheat program, because PH1 is the major responsible factor in order to enable the hybridization, and that, or else, in other words, we can say this will enable the suppression of PH1 will enable the transfer of genes either through backcross program or any other hybridization program in order to incorporate the resistant genes from wild species or to cross wheat with other related genera or other species.